Okay, so today we're going to talk about genetic engineering, which is the next topic that you are going to be exploring in your lab. So before we do that, I wanted to give you a little background into the history and the process that's involved. Essentially what genetic engineering is when you insert some sort of foreign gene of interest into a host cell, and then you use that host cell to transcribe and translate a protein you're interested in. Now, this was first done in the early 1980s to produce insulin. Those of you who know people who are diabetic know that they need to inject themselves with insulin. It was figured out in the early 1900s that insulin could really help diabetic individuals, so the problem is they didn't have a source of insulin. They used to actually have to harvest it from cows or pigs. But in the early 1980s was when they figured out that they could actually take the gene for insulin from a human cell, put it into a bacteria, and then use that bacteria to do the processes of transcription and translation to create human insulin. So what we're going to talk about today is how do they do that? What are the different parts of the process and the tools involved? The general steps are the following. You're going to get the gene you're interested in from the host individual, from the original source. In this case, let's say the insulin gene. Then you have to find a way to insert that gene into a host cell, for example, bacteria. And then once you've done that, ideally you should just allow the host cell to multiply and express or transcribe and translate that gene for you. And you'll get your desired protein, in this case, insulin, right? So nowadays when diabetics inject themselves with insulin, it is produced through genetic engineering. It is human insulin that's been produced in bacteria. And you also get lots of cells that essentially become your protein producing factory. You get all these cells that now contain the human insulin gene and can make human insulin in bacteria. The big picture then is that we're taking the inserted gene, we transcribe it, translating it using the resources in the host cell. So the RNA polymerase, the ribosomes, all the different building blocks. And so we're going to talk about how we do that today. To give you a little bit of background, bacteria have these things called plasmids. So if you look in the picture here, you can see that they have their own chromosomal DNA. That's sort of the instructions on how to just have the bacteria function. But what bacteria also have is these little tiny circular DNAs called plasmids. And they're what we consider extra chromosomal. So they're bonuses. They are not essential for the E. coli or the bacteria to survive. They're not part of the genome itself. They're extra. So kind of like on your car, right? You need your motor and you need your wheels to run the car. You don't need to have a radio. You don't need to have power windows. You don't have to have a sunroof. So the plasmids contain a few non-essential genes, the ones that code for these bonuses, these extras. And since genes code for proteins, then if an E. coli has or bacteria has a plasmid, that can actually give the bacteria additional traits, which can be positive or negative. It kind of depends on what kinds of genes are on the plasmid, right? So some of you may have heard in the past there were some things like E. coli scares with food in Jack in the Box. It was a long time ago, I think, actually before you guys were born. But there was a bacterial toxin that was found to be in some of the hamburger meat, and the reason that bacteria was so toxic was not because the bacteria themselves were toxic, but more because they actually contained a plasmid that produced a toxin that then was really harmful to the humans who ate the meat. More recently, I think there have been other examples where I think there was some contaminated spinach with bacteria. Same idea. What makes the bacteria harmful in this case is not the genome, not the E. coli DNA itself, but those plasmids. Whatever extra genes it might have, sometimes it actually can make the E. coli or the bacteria produce toxins. Plasmids are natural for bacteria. It's how they can exchange genetic materials. So it is possible for bacteria to exchange plasmids through a process through conjugation. It's basically where there's cell-to-cell contact and they basically switch, or they can exchange plasmids. but we're not going to really get into conjugation now. I just wanted to mention what it was. Plasmids are naturally occurring. That being said, scientists have found ways to take a plasmid that's naturally occurring and tinker with it so that they can now have it produce a gene we're interested in, like insulin. So plasmids can now be modified in biological labs, and in a later lab we'll talk about how. For now, all I need you to know is that a modified plasmid is what we call a recombinant plasmid, because we've recombined the DNA in a way that's not usually found. And so plasmids can be used as what we call a cloning vector to get the recombinant plasmid into E. coli, meaning it's a way for us to get the gene we're interested in into the E. coli. So generally speaking, a cloning vector, plasmids being one example of them, is just a way to get the gene I'm interested in into my host. In this case, my cloning vector is a plasmid, my host is going to be E. coli. The way that you're going to get the plasmid into the bacteria is a process called transformation. So it's the process in which foreign DNA, in this case the plasmid containing our gene, is physically inserted into our host E. coli cell. Once that E. coli takes in the plasmid, that recombinant plasmid, we call it a transformed cell. Actually, let me go back for you for a second, just in case you need to write that down. All right, so how are we going to get the plasmid into the E. coli? There's several steps. First, of course, we have to take those recombinant plasmids and the host E. coli, we have to mix them together. Because if they're not in the same test tube, of course, there's no way they're ever going to come together. Second, we're actually going to add calcium chloride solution, sometimes called transformation solution. Calcium chloride is going to be helpful because it dissociates and it becomes the calcium and the chlorine ions. And the calcium ions in this case are the important part. Remember, DNA is negatively charged, so that plasmid DNA has a negative charge on it. The calcium ions, what they can do is they can actually neutralize the negative charges on the plasmid DNA. And that's going to be important because also that's going to help the plasmid enter the membrane. Because also remember that the phospholipid bilayer, phosphates, right, are also negatively charged. If you take two negative things and you try to put them together, they're going to repel. So naturally, this plasmid can't even approach the cell membrane without getting repelled because there's a negative charge on the DNA and there's a negative charge on your phospholipids. So first of all, by adding calcium chloride, you're neutralizing those negative charges. So that's half of the battle, right? They won't get repelled. But we still need to open up some sort of opening in order for those plasmids to get into the bacteria. And that's where the second step comes into play. Next, we're going to do something called heat shock. You're first going to put the bacteria and the plasmids in a really, really cold temperature in ice. And then all of a sudden, you're going to heat shock them. You're going to put them in a really warm bath. By rapidly changing the temperature of the solution, what happens is temporary pores or holes open up in your cell membrane. And when that happens, that provides the door. That's the opening for those plasmids, if they're nearby, to slip into the E. coli and essentially now be transformed. So the heat shock's important because now that we've neutralized the repulsion between the plasmids and the membrane, now we want to create an opening for those plasmids to enter the E. coli. So by going from cold to hot to cold again, we're temporarily opening up some holes for the plasmid to slip in. Now it's important to note transformation is not 100% successful. And what I mean by that is we're assuming certain things that may or may not be true, right? We've put in the calcium chloride, we've added the plasmid, But at the moment at which we heat shock it, if there happens to be a plasmid nearby that can slip in, it'll do so. If there's not a plasmid nearby, those holes will open up in the membrane, then they're going to close right back up again. So it is entirely possible that some of the cells that go through the whole transformation process don't end up with a plasmid in the end, simply because there just didn't happen to be a plasmid nearby, right? Kind of like if I was to throw candy out into an audience, or, if you don't happen to be near the candy, you're not going to get candy, not just because someone threw it out there. So after transformation, some cells will contain the plasmid, and we call those transformed, and then some cells still won't contain the plasmid, and they will be untransformed. And we'll talk about later how we figure out, how we distinguish between who got the plasmid and who didn't. But it's important to note again that just because you did the transformation process doesn't mean every cell actually got a plasmid. In a later step, we'll talk again about how we figure out which cells were transformed and which ones remained untransformed. Now, the last thing I want to talk about with this particular lecture is how do we pick the hosts we pick for genetic engineering? So in our lab, we're going to be using E. coli, very standard bacteria. And why do we pick it as a good host cell for genetic engineering? A couple reasons. It's a good host cell, number one, because in a lab scenario, you want to have fast results. And so E. coli actually reproduce really, really quickly. They reproduce once every 20 minutes. So if I started with one E. coli, after 20 minutes I'm going to have 2, and then after another 20 minutes 4, and then 8, 16, etc., etc. After 24 hours, one E. coli essentially is going to generate like millions of copies. So in terms of fast results, that's a really good thing. Secondly, again, certainly some E. coli are harmful, but the ones that we use in this case are non-pathogenic. The strain we use for this lab is not a harmful strain of E. coli. Of course, you should still use standard, lab safety and, don't have any food near your lab bins and whatnot. But the general idea is it's safe. It's not something that's going to harm you or anyone else. Lastly, if we are going to put a gene into the E. coli, it would be nice to know what we're putting it into. You don't really want anything unexpected. So the E. coli genome itself has been fully characterized. We know what all the genes are in E. coli. We know what they do. We know where they are. So when we're inserting something, there shouldn't be any surprises. And that's what we want as well. We don't want some sort of unexpected result. So hopefully that kind of gives you a quick idea into how genetic engineering works. And in the next lecture, we're going to talk about the background of your specific lab. So the plasmid you're going to be using and what you're going to be doing in the lab in order to genetically engineer your E. coli.